This method can help answer key questions about dynamic protein synthesis and the proteome involved in the process. This technique allows identification of the mRNAs that are being selected for translation, the microRNAs that can participate in the process and interfere with translation, and the proteins that contribute to protein translation or are themselves being translated.
This method can provide insight into cardiac responses to stress, but can also be applied to the study of acute responses to stress in other organs.
Demonstrating the procedure will be Miroslava Stastna, a project scientist from Jennifer Van Eyck's laboratory.
On the day of the experiment, fill the left compartment of a gradient mixer with 5 milliliters of 15% sucrose gradient solution and fill the right compartment with 5 milliliters of 50% sucrose gradient solution. Open the tap and switch on the pump to stir the two small compartments with a magnetic stir, and use a tube linked to the second tap to allow the mixed sucrose solutions to flow into a single ultracentrifuge tube with thin walls on ice. Next, homogenize the fresh or frozen heart tissue with a polytron and filtered lysis buffer for 15 seconds on ice. At the end of the incubation, centrifuge the lysate to remove the insoluble material and harvest the supernatant. Set aside 50 microliters of the supernatant for protein determination, RNA isolation, and RNA integrity analysis, and layer 100 to 500 microliters of the remaining supernatant over the gradient solution.